Hello and welcome to this video on how to answer long answer questions on culturing microbes. One question that you may well be asked in the GCSE exam is how microbes are grown in lab conditions. If this is a long answer question, it will follow the following format. There will be a short sentence at the top talking about using good English and organising your information clearly, and it will expect you to use the right terminology and keywords. In this question, it states the student is told to grow some bacteria on agar jelly on a petri dish. Describe how a student should prepare an uncontaminated culture of bacteria in the petri dish. You should explain the reasons for each of the steps you describe. So, as this will take the form of a 6-mark question, there are a few important things that you must remember if you are to gain all of the marks. You must make at least 6 points. You must remember to write in full sentences. When answering this question, it's worth 6 marks, so realistically you should be trying to answer it in about 5 minutes. To give yourself a minute to quickly check through it and make sure that you've not left anything out. It's also important to remember to use the key terms. The first stage in answering this question is to talk about setting up the Bunsen burner. This is to kill any bacteria that are floating down in the air as you're working. Some key points to include at this stage is that the Bunsen burner only needs to be on a safety flame. This is to ensure that it covers the maximum possible area and kills most fungi that are also dropping down. It's important to talk about constantly working underneath the flame. This is an area that people often leave out and lose marks for. And as always, throughout this question, you'll need to talk about how the different stages stop contamination of your culture. The second stage to talk about is sterilising your inoculating loop using ethanol. Some key points to remember is that sterilisation means to kill 100% of the bacteria. You need to remember that killing off all the bacteria stops contamination. You also need to mention not to leave the inoculating loop in the flame for too long. If you leave it in the flame for too long, then you run the risk of going and it being too hot when you're placing it in the bacterial culture and it killing all of the bacteria. Stage 3 is talking about going and transferring the bacteria onto your agar plate. Some key points to mention are that you must let the loop cool down or else it will kill all of the bacteria. You can spread the bacteria in lines as this will help lower the concentration of the bacteria and allow you to see individual colonies of bacteria growing. And it's important to talk about not pressing too hard onto the agar as this would cut into it and would make it more difficult to physically see any colonies growing. After you have gone and used your inoculating loop, you've got to talk about sterilising it again, either by running it back through the flame or putting it in disinfectant. This is important to help stop any future contaminations. You need to talk about sealing the lid of your agar plate in 4 different places, but not all the way around. When sealing it in 4 different places, this allows oxygen to get to the bacteria, which means that aerobic bacteria will grow, as anaerobic bacteria is dangerous because it could grow inside of us. The final part in your answer needs to be how you're going to allow the bacteria to grow. You allow it to grow at 25 degrees for around 3 days. 25 degrees is used because the bacteria will grow at a reasonable rate. In industry, you would use a high temperature to speed this up. However, we do not want to grow bacteria at 37.5 degrees, as this is body temperature, and if you breathed it in, it could be harmful. One final thing that they quite often like to chuck in to questions about growing bacteria is something about a diagram that shows a lack of bacterial growth. So, the darker areas show where the bacteria have grown. The lighter areas around the black discs show the area of no bacterial growth. This must mean that there is something on the disc that is stopping the growth. Therefore, the larger the area, the better the antibiotic or disinfectant will be working. So, to finalise, in your question, it's important that you're covering the following 6 key points. Sterilising your loop, taking the bacteria sample, inoculating your plate, re-sterilising the loop, allowing your bacteria to grow under safe conditions. I hope that you found this helpful, and there is a link to the 6-mark question in the comments below. Good luck.